This is the second of 3 videos in Module 2, DNA replication. In this video, I will specifically be talking about the properties that are unique to replication of prokaryotic DNA, or DNA in a cell that does not have a nucleus. Bacteria and viruses replicate their DNA uniquely compared to eukaryotes, mostly because of the structure and shape of the genome. Bacterial genomes and viral genomes are very small, and they're also circular. And so there are some properties that some factors that bacteria and viruses can sort of get away with because of their circular small genomes, where eukaryotes have to come up with unique workarounds for those issues. And so the mode of replication depends on the structure. When we talk about a replicon, this is a region of DNA that replicates from a single origin of replication. And so when we talk about bacteria replicating their genome, a new genome will be referred to as a replicon, since that was initiated from a single origin of replication. An origin of replication is a particular sequence in the circular genome where replication begins. And it can be bidirectional, as is the case with the theta model of DNA replication, or unidirectional, as is the case with rolling circle DNA replication. Because the genomes are circular, we have 2 different types. Again, theta replication, which is a bidirectional replication initiated at a single origin of replication. And so a single origin initiates the start, the bubble will open, and then 2 replication forks will proceed in opposite directions. Rolling circle replication also begins with a single origin, but DNA replication begins and moves in only a single direction. And so this is only efficient if the genome is very small. And so this is reserved mostly for viral genomes and in plasmids, which are smaller versions of chromosomes in bacteria. Theta replication is a type of replication common in all bacteria that have a circular DNA. So all bacteria, and they also have to be contain no nucleus. And so let's walk through this. Here's our double-stranded DNA, our regular parent strand. The origin of replication is a single spot where DNA is replicated. So initiation begins here. A replication bubble forms, which is basically a gap between the 2 double-stranded pieces of DNA where hydrogen bonds have been broken, and so we're separating the 2 strands of DNA. And this is what the replication fork looks like. So this is the old strand, the old double-stranded piece, and these are the new strands that are being apically replicated at the fork. The fork will then proceed around the circle, moving towards itself, right? So they're moving away from each other, and then eventually they will join at the end where the 2 circular DNA molecules are joined. To get rid of, or to solve this problem of this entanglement, an enzyme will cut, called gyrase, will cut these pieces, and then ligase will stitch them back together then, eventually producing 2 circular DNA molecules. And so we can see this in an EM image. So here's our EM, or electron microscopy image of a bacterial genome. And we can see the replication bubble forming and then 2 replication forks. We can then see 1 new strand coming off. And so we're proceeding in this direction and this direction of the fork. And then slowly those 2 forks are going to meet up with each other right here and then split up as is seen in this picture. This right here is what's called the theta shape, or the theta formation, because it looks like the Greek symbol theta. Rolling circle replication is only reserved for very small genomes. So some viruses, so not even all viruses, but some viruses that have a circular genome, and for some E. coli plasmids, specifically the F factor. But other types of plasmids will also do this. And so replication is initiated not at an ORI, but just by a simple break in one of the 2 nucleotide strands. DNA synthesis will begin at the 3' end of the broken strand right here. And then the inner strand is used as the template. And then so the new strand will slowly displace the old parent strand until it bumps into itself. Cleavage will then release a single-stranded linear piece of DNA and then the double-stranded circle. So we end up with this, and then we end up with a piece of single-stranded old parent DNA that will recircularize, and then a new piece of DNA will form there. So still following the semi-conservative model. This process is a lot shorter, though, than theta replication because we're only moving in one direction. So again, because this is a longer process, rolling circle replication takes longer. It's only really efficient if the genome is small. Theta replication is much faster. Bacterial replication requires a good amount, a large number of enzymes and proteins to make it happen, and it proceeds in 3 different stages: initiation, elongation, and termination. So initiation refers to the process of binding to the origin, opening up the double-stranded piece of DNA, and stabilizing that interaction. Elongation represents the stage at which polymerase or DNA polymerase is creating a new strand of DNA. And termination is the signal that tells DNA polymerase to stop making a copy. And so let's go through each of these 3 stages. Initiation starts at the origin of replication in bacteria, called the ORI, or the ORI-C, depending on the species. But it's always ORI, or ORI, and the origin of replication. It's usually about 245 base pairs long. An initiation protein is what recognizes the ORI. So these initiation proteins, particularly one called DNA-A in E. coli, is a DNA binding protein, and so will bind to ORI-C based on its specific sequence. And then unwinding will occur. That's initiated by this DNA-E or initiator protein, and then continued by the enzyme DNA helicase, which travels down the core of the DNA double helix, breaking hydrogen bonds. Single-stranded DNA binding proteins will bind to the single-stranded DNA, making sure that it stays that way and doesn't just snap closed back on itself. And then DNA gyrase removes some of the overwinding that occurs as we open up a bubble in the middle of a circle. And so let's see what that looks like in picture form, because it's a lot easier to see in pictures. Okay, so here's the ORI-C of E. coli, 245 base pairs long, and it's a specific sequence that initiator proteins that bind to DNA will recognize and form this loop. And so now here we have the DNA-A initiator proteins bound to ORI-C. This causes a short stretch of DNA to open up at the end of this loop, at the base of this loop. And that tiny little spot represents an open window of opportunity for helicase to come in and attach, and single-stranded binding proteins to attach to keep this replication bubble open. DNA helicase then binds to the lagging strand template at each replication fork. So we're binding to the lagging strand. If it bound to the leading strand, it would bump into the newly synthesizing DNA all the time. So it has to bind to the lagging strand where there's no DNA being created. And it sort of unwinds and moves and chases the replication fork. So we're chasing the replication fork, unwinding the DNA as we go, breaking hydrogen bonds in the core. Single-stranded binding proteins will bind to the single-stranded DNA, preventing it from snapping back closed again. And then if you take a circular piece of a circular molecule and open it up like this, you're going to create a lot of strain down here on the other end where it's still double-stranded. And so DNA gyrase will bind and get rid of that strain so that the molecule doesn't become too unstable. Elongation is the process of creating a new strand, a new DNA strand, using the original DNA strand as a template. This requires a 3' OH group on the new strand. But as you can see here, when you open this up, there is no 3' OH group with which to begin. It's totally naked, right? There's nothing bound to it. So an enzyme called primase binds to the area of the DNA, so binds to single-stranded DNA, and creates an RNA complement for the DNA template. And so a small stretch of RNA molecules, about 10 to 12 nucleotides long, is created by primase, and that's represented by this pink stretch with the start flag right here. And so this represents a free OH group with which DNA polymerase can bind and add new nucleotides. So it has to be added anti-parallel. So here we have our 3' end of our parent. So the primer is added anti-parallel to that, starting with the 5' end, and then built. And primase does not require an OH group to start making RNA. Only DNA polymerase requires that. And so DNA polymerase will bind here and then start attaching and then chase that replication fork. Gyrase is over here unwinding on the lagging strand, and then primase is also building primers so that there's a free OH group on the lagging strand for DNA polymerase to elongate. So here's this primase, here's this in action. So here we have gyrase. This is relieving some of that stress caused by the unwinding. Here's helicase on the lagging strand, making sure that hydrogen bonds are broken and this replication bubble is open. Then we have our primase, and that's creating a new RNA piece of RNA. So primase synthesizes short stretches of RNA nucleotides, providing an OH group on a 3' carbon so that DNA polymerase can then bind and add new nucleotides. And so these green segments are primers. They're RNA. And so initially, at first, when DNA is replicated, it's a hybrid of DNA, which is shown in red, and RNA, which is shown in green. Oops, sorry. On the leading strand, we need only one primer, right? So it's a continuous replication where we chase the fork. So we need one primer here, and we're going to chase it in this direction towards this fork. The leading strand, so this is the leading strand for this replication fork. This is the leading strand for this replication fork. The lagging strand, however, is going to require multiple primers be made. And so on the lagging strand, replication is discontinuous. And so we have to continue generating new fragments of DNA, which means we're going to need multiple sites, multiple OH groups for DNA polymerase to bind to. And so we create a primer here and elongate. So actually, this was the first one to be made and elongate. And then we make this one once we have space and elongate. By the time we get here, this is open. So primase makes another one and elongates. And that's why on this other image, primase is shown here very worn out and tired because it's continuously making primers on the lagging strand. Elongation, or creation of a new DNA using an old DNA template in bacteria, is carried out by DNA polymerase 3. After DNA polymerase 3 has synthesized all the new DNA, we now have that weird hybrid of DNA and RNA. And so the RNA primers have to be removed so we don't have that hybrid. That hybrid is not stable. So DNA polymerase 1 comes in and chops up and removes the RNA primer. And then DNA polymerase 3 will come in, bind to the OH group at the beginning of that gap and fill it in. After the RNA primers are removed and then DNA polymerase 3 fills in the gap, all the hydrogen bonds that are going to be made are already present, but there is still one last phosphodiester bond that has to be created, and that is made by DNA ligase. And so that connects all the open gaps in the DNA backbone to make sure the backbone is stable. Termination in bacteria is really easy. When the two forks meet, they just bump into each other and displace each other, or sometimes there's a protein that facilitates that process. Okay. There are lots of different types of DNA polymerases in bacteria, 5 in fact, and they all have slightly different functions. So the major elongation polymerase is this one, DNA polymerase 3. This one is elongating in a 5' to 3' direction. They all do that actually. It also can do it in the opposite direction. And so DNA polymerase 3 also has this property where it can sort of back up a little bit. So if it inserted one bad nucleotide and then maybe got one nucleotide beyond that, it can back up maybe 2 total nucleotides, chop them up, and then continue elongating. So it can sort of be making DNA in this direction, back up a tiny bit, oops, I made a mistake, and then continue. DNA polymerase 1 also has this ability. This is actually needed in order to remove and replace primers. And it also has this ability to remove incorrectly paired nucleotides, or does a little bit of DNA repair. 5' to 3' exonuclease activity is reserved mostly for just DNA polymerase 1. Oh, this arrow should be pointing here. Sorry about that. Okay. And so, sorry about this error. I'm going to correct this now. Okay, sorry about that. And so DNA polymerase 1 has this other unique property that the others don't have, and this allows it to remove and replace primers. And so these three, 1, 2, and 3, have this ability to correct DNA in the opposite direction that it's made. All of these polymerases have the ability to polymerize DNA in the appropriate direction. Some of them are involved in DNA repair. Some of them are simply elongators. Some of them remove primers. And so each have unique functions and some have overlapping functions. I would ask you to please understand the function of the ones that I described on this slide, DNA polymerase 3 and 1. Okay, so here we have the very last part of DNA replication in a bacteria. So here we have a fully synthesized new strand. So here's our template strand. Here's the 5' end and the 3' end. It was synthesized in the opposite direction, so in the 5' to 3' end. We needed an OH group to start with, and so first an RNA primer was put in place, and then DNA was added using this free OH group at this end of the RNA primer. And so we were able to make this DNA. But now we have this RNA causing a problem in there. We really need to get rid of that because a hybrid molecule of DNA and RNA is not stable. It would fall apart. So DNA polymerase 1 will bind and start chopping up the primer in a 5' to 3' direction. So it starts here at the 5' end and just gobbles it up, removes it completely. Upstream of that is the other end of the circle that's sort of coming in to complete this process. And so when DNA polymerase 1 removes the RNA nucleotide, because this is a circular genome, the other end of the replication has caught up. And so we now have a 3' end that's open, and DNA polymerase 3 can come in and fill in the gap. And so we have all of our nucleotides in place, all the hydrogen bonds are in place. The only thing we have to do is create that last phosphodiester bond. And so that is done by DNA ligase, which seals the break with a phosphodiester bond between the 5' phosphate group of the first nucleotide that was added, and then the 3' OH group of the last nucleotide that was added by DNA polymerase 1. And so DNA polymerase 1 can fill in this gap. DNA polymerase 3 can also fill in that gap. And then ligase creates the last phosphodiester bond. And so here are all those enzymes, all summarized on a table right here. So we have our initiator proteins, helicase, single-stranded binding proteins, and gyrase. These are all part of the initiation phase of DNA replication, and each have their own unique property. Then we have the elongation phase, which is started by primase, continued by DNA polymerase 3, and then sort of all the cleanup job done by DNA polymerase 1. And then termination happens on its own when the two forks bump into each other and is completed by DNA ligase, which makes the last phosphodiester bond, finally creating the double-stranded new piece of DNA.